Yes, ma'am. It
Sorry, I
started
just
now.
I just started. Okay, okay, let me just say it in brief. So Arthur Kornberg's lab was the first to do this because they were biochemists and they knew how to work with nucleotides like DNTPs, NTPs, even NAD. They were working with these. And so the first thing that they were trying to do is they had an idea that if something is going to help in polymerizing nucleotides, then it must be an enzyme. So that was a firm belief for all these biochemists who believe that biological reactions have an energy barrier that must be lowered with the help of enzyme. So they were searching for enzyme that can help in polymerization. And they took E. coli cells because E. coli cells have a very fast doubling time, which is they double in every 20 minutes approx. So they took these E. coli cells in the hope that if they have to double in 20 minutes, then they will have polymerases that can help to do this reaction. And so they burst open the E. coli cells. So if you burst open the E. coli cells, you get all of these proteins in the supernatant. They will be of different sizes, right? Now, if you are able to separate out the proteins based on their size by some chromatographic method that is able to release the larger proteins first, then the smaller ones, then the tiny ones. You are able to separate out the proteins based on their size, hydrodynamic volume. You're able to do that. Now you test each of these fractions to see which fraction is able to incorporate P32 labeled DNTP into DNA. If it is incorporating into DNA, then even after acid wash, it will remain in a pellet. If it is not incorporated into DNA, then after acid wash, it will be in the supernatant. So that's a very simple assay that they did. And they observed that all 4 DNTPs are substrates for this enzyme that they called polymerase. So they termed as polymerase because it was polymerizing the DNTPs. So if you have a look at this at the reaction at the bottom, so this is exactly what Arthur Kornberg had written in his paper. This comes directly from his paper in 1957 JDC, which shows how they were looking at it. They were literally chemists. So they were looking at it as a reaction where you take DNA and you take these substrates, and once you add this extract, the DNA into the DNA, these substrates get incorporated and the PPI is released. So they looked at the whole thing in terms of this reaction. And in fact, initially Kornberg did not quite have this idea that if you have a template DNA, the nucleotides that are added kind of have to match the template DNA, which means that you are creating a complementary DNA. He did not even have this idea. He was working on the aspect of identifying polymerases. This idea came into his paper much later when he started to look at what Watson and Crick and others had suggested, that one of the strand can act as a template for the other strand. And so then the idea came up that, okay, you can have these specific nucleotides being added to create a complementary strand. That is another reason why when he was adding tritiated DCTP, he only had a very tiny amount of that being added to his reaction. Now, can any of you say why? So he took the E. coli extract, he took DNA, and he only took DCTP, and only a tiny fraction of it got added into DNA. The rest of the DCTP was in solution.
And because there might not be other nucleotides to be added, like after C there would be A, or my C, or
Exactly.
it would be T.
Exactly. Exactly. So because he was not giving the other 3 nucleotides, only a very tiny amount of it was getting added because it is a template strand which is guiding the synthesis of the second strand. And so if you do not give the other 3 nucleotides, after a while, maybe 1 or up to 2C, it just stops, or maybe even 1C doesn't add to depends on the template DNA that he was using. So in the end, if you put together all of this, he was able to identify a few things. One is that all four nucleotides are the substrates for this enzyme. He called it the polymerase, and that it requires this guiding template strand for synthesis. And he even went on to identify the enzyme in the fraction, and he called it the DNA polymerase. Now, what he did was he did not identify DNA polymerase 3 as Ratnesh or I don't know, Sarika. Somebody has explained that there are 3 DNA polymerases. In fact, there are more than that in E. coli. But 3 DNA polymerases, 1, 2, and 3, 3 being the one that is responsible for replication of the chromosome, 1 being the one that is responsible for sealing of these Okazaki fragments by removal of the primer and also in repair synthesis. So he did not identify 3, but he rather identified 1. So after Arthur Kornberg and colleagues, they identified the DNA polymerase. You can see that there has been a burst of identification of polymerase. So somewhere in 1956 is the time when they identified the DNA polymerase. You see the identification of mammalian polymerase, mitochondrial polymerase, 2 other DNA polymerases, telomerases. Then it goes on, including discovery of reverse transcriptase and discovery of other DNA polymerases, even in viruses, bacteriophages, and so on. So we now have a host of DNA polymerases that are available to us, RNA polymerases that are available to us, but there are some despite the huge diversity, despite the fact that some of them are single protein, some of them are multi-protein complexes, they all have a few common features. So what are the common features? The common features actually have already been discussed by you, but we'll just go through it quickly. So they all require a template DNA. They all require these 4 NTPs or DNTPs, whatever is the case for DNA or RNA. They require a directionality, and some of these polymerases have something called a exonuclease function. So not all polymerase will have exonuclease function, but the DNA polymerase 1 that we will discuss today has exonuclease functions. So it has a 3' to 5' exonuclease activity called proofreading activity and a 5' to 3' nuclease activity required for editing. So can anybody explain what exactly is meant by 3' to 5' exonuclease activity?
Ma'am, basically DNA polymerase, what it does is after adding this base, it checks whether it is correctly added or not, and if it is correctly not added, it removes that, like in 3' to 5' direction, and adds the appropriate nucleotide. So I guess
So
that is
basically,
what.
after adding the DNA nucleotide, it has to translocate in front, but it can also check with one of its domain if the added nucleotide is correctly added. If not, it can remove it, go and add back another nucleotide and proceed. So basically, it translocates DNA, but it can also do this cross check to see if it has added a correct nucleotide. You're absolutely right, Ratnesh. What about 5' to 3'?
Ma'am, it is for primal removal, editing for removing the primer. So in the direction of 5' and to 3'.
Okay, okay. All right. All right, thank you. So we will now look at one. So I think this is again something that comes directly from your textbooks, and you've all perhaps seen this figure or read it sometime. So when we talk about DNA replication, the idea is that DNA replication doesn't start anywhere in the DNA in the chromosome, but it requires specific regions for DNA replication to start, which are called origins of replication. And I'm sure you have already read this. So when they identified first the origins of replication, the idea was from the origin of replication, does the replication go only in one direction or is it bidirectional? So here is a beautiful example through a pulse chase experiment. I'm sure you have all read it, but I decided to add this to your slides just to emphasize that if you're trying to look at how a DNA polymerase works, then there are multiple ways, multiple experimental methods that you could use it. So what is a pulse chase experiment? So suppose you take bacterial cells, human cells, any cell that you would like to study, and you put that into a radio-labeled nucleotide soup. So just like Kornberg did, you put it into radio-labeled nucleotide soup. So every time the DNA synthesis is occurring, it's going to add radio-labeled nucleotides. Now if you take it out, if you take out those cells and put it into a media without radio-labeled nucleotide, just with normal nucleotide, then after some time inside the cell, you would have lower levels of these radioactive nucleotides and higher levels of normal nucleotides. So initially you would have radioactive nucleotides being incorporated at all the steps, and then you have changed the soup. So now it has these normal nucleotides being incorporated, but once in a while, one or two radioactive nucleotides might get incorporated. So now if you have this condition and you're looking at it under an autoradiogram, what is it? You burst open the cells, take out the DNA, and you take an impression of the radioactivity on a film. If you do that, then you would be able to see spots on the film wherever the radioactive nucleotides are present. That's autoradiogram in very simple terms. So if you do that, initially when the cells were growing in radioactive nucleotide, you had all these radioactive elements being added, one after the other. So you would see a patch of radioactivity, a patch of dark spots, very closely spaced dark spots. But once it has been pulsed, which means once it has been removed and put into normal nucleotides, not radioactive nucleotides, normal nucleotides, then like after a few steps, maybe one radioactive nucleotide would be incorporated. So you will start to see discrete dots after that. So these discrete dots, if the replication is moving in one direction, would only go in one direction. But if it is a bidirectional replication, you will see those dots occurring on both the ends. So now if you try to look at this image, you see these closely spaced dots, and then these sparse dots are in both ends. So it is here, both ends, which indicates that from the origin of replication, replication can proceed from both directions. So it's bidirectional replication. So something that you've already learned. I'm just presenting this image once more. So here are certain things about DNA polymerase 1. The reason I've put this is because the next few slides would require this information. So if you look at DNA polymerase 1, it's considered to have a thumb, finger and palm like structure, in the sense that the palm is the place where there is active site for synthesis of DNA, that is a polymerizing reaction. And the thumb would guide the exit for the newly synthesized single-stranded DNA strand. So the newly synthesized strand. But apart from this, it has another additional domain. The additional domain will help it to have a 3' to 5' exonuclease function, and another additional domain that will have a 5' to 3' exonuclease function. So if you remove this 5' to 3' exonuclease function, you create something called the clinofragment. So you must have heard about clinofragment. Clinofragment is nothing but if you take DNA polymerase 1, you digest it with subtilisin, then you end up with DNA polymerase that lacks this 5' to 3' exonuclease activity because you have cleaved off one part of the polymerase, and it still has the proofreading function, but it lacks this 5' to 3' exonuclease activity, has the polymerase function, that's the cleano fragment. So what is it? So this DNA polymerase 1 will have a certain speed of replicating DNA. Now, how do you find out what is the speed of replication of this DNA? And this experiment here describes to you this. So consider, so the experiment is really simple as it can get. So consider a rolling circle DNA replication. Now, have you heard of rolling circle DNA replication?
Yes, ma'am.
What is it?
Ma'am, basically in this the replication like after cleaving at origin of replication, the replication starts like that fragment is starts protruding from this circle, and the polymerase only replicates uh, the fragment which is being uh, 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 like ejected from there. Is something like that.
Okay, anybody else would like to go for it?
Ma'am, it is the mode of replication in plasmids or for some bacteriophages. So in this, what happens is that for the two, it is like two strands in circular form, which are complementary to each other. These are nick or produced in one strand by an endonuclease. So for the 3' end of this, where the we have produced the nick, is then uh, is then act as a primer, and it add nucleotides and it synthesize the new strand complementary to the other strand it has originally have had.
Okay, I partly correct. The idea of rolling circle replication is that suppose you have the circular. You need a circular DNA for this. Rolling circle replication cannot work with linear DNA. If you have a circular DNA and you are starting to synthesize this complementary strand, then once it completes it, it needs to stop here. What if it does not stop, but it keeps on going round and round and starts replicating this DNA multiple times? That is what is going to give you rolling circle replication. And if rolling circle replication works, you would end up getting very long stretches of these catenated DNA. So in this experiment, they have utilized this because you would be able to see this DNA synthesis keep going on, and you get very long stretches of DNA. So first thing that they have used here is rolling circle replication. They have not taken linear DNA, they have taken circular DNA, where if you start replicating from this end, so say suppose you've added a primer and it's replicating from the primer, and then it can start to go past the primer. You kind of push out the primer, and then you start replicating this DNA again. And every time you keep replicating this single strand of DNA, remember it's happening for both the strands, but you keep replicating this strand of DNA, the linear strands that you have produced are already combining with the existing strand, but you keep moving on with it. You have to add the helicases, add everything that is required for replication. And you need to have those ideal conditions for this to occur. Without helicases, this will not be possible. So if you need to have a rolling circle replication replicated in silico under a glass slide, you need to give the system all the proteins that it needs, including helicases. You need to provide it the primer, the DNTP, polymerases, of course, anything else that holds it together. If there are clamp proteins, you need to give it the clamp proteins. Everything that it needs has to be provided so that the rolling circle replication takes place. Given that you have given all of these proteins and you have the right conditions, DNTPs, pH, etc., you will be able to observe rolling circle replication in on a glass slide. You do not have to do it inside a cell. So they took the same mechanism to a glass slide. So here you can see an additional thing that they did. They managed to attach this circular double-stranded DNA to a glass slide with the help of streptavidin biotin. Now this chemistry has already been discussed the last time by Dr. Saurabh Raj. He explained how you can use biotin and streptavidin to attach pieces of DNA to a glass slide that you can then use to pull using a magnetic tweezer. So something similar you can do here. You attach these rolling circle replication substrates to a glass slide. Alright, done that, you add NTPs. Now how do you follow the occurrence of this DNA replication? You need something to read, right? In the previous cases you have seen that in 1950s they were using radioactivity. But nowadays it's much more easy to follow these using fluorescence. So you've all heard of ETBR, ethidium bromide.
Yes, ma'am.
Okay, what does ethidium bromide do?
Ma'am, when we have to like unwind this DNA, so ETBR goes into the uh, 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 clips of this DNA, and when it goes there, it what
No,
it
no,
does.
no.
Yes, ma'am.
Ethidium bromide does not cleave DNA. Wait. You're
No, ma'am.
going all over.
No, ma'am. I am telling. So after it attaches to this DNA, we can use this UV light to see this DNA. Like it has some kinds of
Okay.
activity.
Ethidium bromide is a planar ring-like molecule. Because of its planar ring-like structure, it can go and intercalate. What does that mean? If you have these base pairs, it can go and stack between the base pairs. And this stacking gives it certain electronic configuration that helps in its fluorescence. So if it is not stacked, it will not emit the fluorescence. But when it is stacked inside DNA, then it starts to emit a fluorescence. That's the reason you are using UV light to excite it and see fluorescence. So similarly, you could use other structures, other chemical compounds with these ring-like structures that can go and intercalate. What does intercalate mean? You have these two base pairs. इनके बीच में जाके अगर वो घुस जाता है, then it goes into an electronic configuration that is going to help it to fluoresce. Otherwise, these molecules in solution will not fluoresce. If you add something like that, you will be able to follow rolling circle replication. So in this experiment, they did something very similar to this. They used Cytox orange to stain the double-stranded DNA. Now, they used two different kinds of circular DNA to observe this kind of replication. One is the DNA from T7 bacteriophage. One is E. coli replisome. So all they use two protein machineries to do this. One is T7 DNA replication machinery. One is E. coli replication machinery. They used both of these, and then they studied they followed the formation of this rolling circle DNA replication. And is the video I'm going to show you. So this is T7 DNA replication, and you can see that, okay, how are they able to see rolling circle replication as a single line? So they did something else. You have rolling circle replication occurring, so you should be able to see circles of it, like it should be all jumbled up. But suppose you have put this in a flow chamber, so there is a liquid flowing in one direction. So all of these replicated DNAs would start to flow in that direction. So you would see double-stranded DNA being pulled out in that direction. So it's a very simple concept, but it's unique. So you see these rolling circles occurring. Now, because there is a flow, suppose there's a flow of water, so they would start to go in that direction and they will be pulled in that direction because of the flow of the liquid medium. So they use this concept, as you can see here. There is a flow that is causing the DNA to, the rolling circle DNA to move in one direction, and that can be visualized very clearly under microscope. What kind of microscope? Any ordinary fluorescence microscope? No. You need turf. Why turf? Because you would like to see events that are occurring very close or at the surface of the glass slide. You want to just excite the events occurring at the surface of the glass slide, not throughout. If it is happening anywhere in the bulk solution, any noise there, you want to cancel it. So you have very crisp images. So again, they have used turf. I've already explained turf. If anybody has any confusion, I can explain it once more. Is turf okay? No?
Yes, ma'am.
No, no questions? Okay. So they have
So,
used
ma'am,
turf to do this.
I have a question. So, ma'am, why exactly ethidium bromide is needed for what is called to visualize this DNA? Like somewhere I've read like while what we do is while denaturation of DNA, we can see this like without using even like just in water, and we can see using UV light
What you can see.
this DNA.
You can see it in water in.
Like when DNA denatures and like we try to see like how much it has been denatured to at that time.
You can see that where?
Like in water.
How? How did you follow it? There are
Using
multiple
UV
ways
light,
by
right?
which you can follow denaturation of DNA. The question is how, where did you read it and how was it followed there?
Oh, perhaps I will have to read again.
Not perhaps, you definitely have to read it again. See, if you are, so that is going to take me off the class completely, but I will do it if you have 10 more minutes after the class. So, for the moment, let's consider that this is using turf microscopy, you will be able to follow the replication of this rolling circle by looking at how much of this DNA is getting elongated. Why it is getting elongated? Why not jumbled up? Because of a flow. So they have added a flow. Every time the DNA is replicated, because of the flow of the medium, it gets pulled in that, simply gets pulled in that solution. So you'll be able to see lengthening of this DNA. Now, lengthening of this DNA, T7 DNA versus lengthening of this DNA with the help of E. coli replisome, DNA polymerase 1, and you can see the replication here moves much faster compared to T7 DNA. And this is E. coli DNA polymerase 1. You see the replication is much faster, much longer. So they tried to quantitate this. Now, quantitation is where all the trick lies. How would you quantitate? You can see now that the double-stranded DNA is getting pulled because of the flow. You can see the rate at which it is getting pulled, how fast it is moving, how far it is moving. But how do you calculate this and come to the point where you can say so many base pairs has been produced in so much time? So to do that, it's a very simple trick that they used. They used a known DNA with a known number of base pairs, and they observed that under the same microscope. And they took an image. And so an image is made up of pixels. So then they calculated how many pixels of this DNA is, or how many pixels is composed in this DNA. So now they know number of base pairs equals to number of pixels. So from there they calculated 1 pixel is equal to how many base pairs. So it's a very simple thing that they did, simple to understand. Suppose you're looking at me on on this camera, and you know that for this camera you have so many pixels is equal to so many centimeters, so you'll be able to get an approximate idea of my height. So something similar to that was done by them, and that gave rise to this graph. So what does this graph say and why is it important? Why am I explaining this to you? Is because you can see that E. coli, there is the distribution, there is a clear distribution of the rate at which the E. coli DNA polymerase 1, because they used only DNA polymerase 1 here, works. So the rate on an average is about say 500 base pairs, but can be as high as 1000 base pairs added per second. So this gives you an idea how fast the DNA replication is actually working. 1000 base pairs per second for DNA polymerase 1, which is still a slower polymerase. DNA polymerase 3 has a much faster rate of replication. And you can see even the lengths at which these replications work, and you can see that they go up to about 400 kilobase pairs, which again indicates how processive these polymerases can be, given that they are working in a replisome. Please remember, they have added 13 different proteins into this. It's not just DNA polymerase 1. Maybe if they had done it only with DNA polymerase 1, the processivity wouldn't have been so high. But given that they have added everything that can produce the clamp, that can hold the DNA, there is the helicase, everything given together, then the processivity can be as high as 400 kilobase pairs. That really goes to show that these enzymes are highly processive and very fast in replicating DNA. So with this, I will stop today's lecture. It was just to give you a preliminary idea of how people have studied polymerases. As an example, I took DNA polymerase 1 as because that was the first polymerase that was ever identified. The entire field of polymerases starts with DNA poly 1. And I've shown you how you can study it, be it radio labeling, because you can study if it is going in a single direction or bidirectional, simple radio labeling and pulse chase experiment. Or you can do a very simple rolling circle replication and you can follow that replication. You do not have to add radioactive labeled DNTPs. You simply use an intercalating dye, and that intercalating dye is enough for you to follow rolling circle replication, given that you have put a flow. So in a flow, this rolling circle replication product would simply sort of flow in the direction of the liquid media and would elongate. So you can see the rate of elongation of this as it keeps forming, as the rolling circle keeps, it's called rolling circle because as the entire DNA gets pulled through the replisome, the circle itself would start rolling and would elongate, and the newly formed double-stranded strand would elongate out because of the flow. It would go in one direction. It would not jumble up. Otherwise, it would have all jumbled up and you would have seen a growing spot. You don't see that. You see this growing end because of the flow. And you can specifically quantify in terms of base pairs how many base pairs are produced per second, because you are able to take snapshots of this event at very fast rate, and then you know exactly how many base pairs is produced per pixel. So you quantitate that and you say that the DNA polymerase 1 is very fast. It can be as fast as 1000 base pairs per second and can be very processive in a replisome. So this is important. It has to have all of these additional proteins with it to produce almost 4000 kilobase pairs at one go. So with that, I will stop today's lecture. If you have any questions, especially Ratnesh, if you have time, I can explain what you ask.
Ma'am, I have a question
Yes.
related to physics.
Yes, yes.
Ma'am, our exam is on 10th April, na? According to the
Yes,
schedule. Okay.
yes,
So you were discussing in the starting, so I thought there is some change or
yes.
something. Okay. And it will be
So
in
your exam will be open book, open source, open internet exam. You will have 3 hours to complete the exam. It will be a short exam of 1 hour, but I'm giving you all additional 2 hours to finish it. And you will have to finish it within the stipulated time that is given in the exam schedule that I will also communicate by email and on Moodle.
and we can give it from our PG home or institute
Anywhere,
anywhere.
anywhere, anywhere, anywhere. It will be not proctored. You can give it from anywhere you are. You do not have to worry about that. You do not have to come to a classroom for my exam. You can give it from any place you are, from your PG, from your home. If you want a classroom, we can also provide you a classroom. If you need a classroom, I'll provide it to you. The only condition is please do not copy from each other. It's an open book, open source exam. खुद से लिखिए, खुद से सोच के लिखिए, ढूंढ के लिखिए, जैसे भी हो लिखिए, लेकिन कॉपी मत कीजिए. Do not copy from each other. That would give you a straight zero.
Ma'am,
Yes, come on.
I have a question about this experiment that Arthur Kornberg did. So, ma'am, in the fractionation, when he looked at the type at the DNA polymerase, and he found that that was the DNA polymerase one. But, ma'am, why the other type of polymerase didn't hinder with the experimental design? I mean, there should be many type of polymers in the E. coli system, no? So why did he pinpoint on a one specific polymerase? I mean, how ignore the other type of polymerases?
Well, he that's a good question, honestly. He did not identify the other polymerases. He ended up identifying only DNA polymerase 1, and it might have something to do with the way his construct was designed. That it was only able to bind to DNA polymerase 1. Maybe his construct was designed in such a way that he was only capturing DNA polymerase 1. But then how people have identified the other DNA polymerases is also interesting. So DNA polymerase 1 is present in quite an enormous quantity in E. coli. So very easy to identify because it's in huge numbers too. Uh, how DNA polymerase 3 was or 2 was identified is also very interesting. So they ended up making thousands of mutations of E. coli just to identify if there is a mutation of DNA polymerase 1, what would happen to the E. coli, whether it will stop growing. And they did these mutations of DNA polymerase 1. They saw that DNA polymerase 1 is no longer present and still the cell is able to replicate. And that is how they hit upon DNA polymerase 3 and DNA polymerase 2. So the one that was the simplest DNA polymerase that they could easily capture with their construct and easily capture with their fractionation methodology was DNA polymerase 1.
Okay, ma'am. Yes, now
But
I get.
it took them a different route to do this. So they could not identify DNA polymerase 2 and 3 by this fractionation method. They had to do something completely different to find out if at all there are other polymerases and then additional methods to do that. So those other polymerases were perhaps not amenable to the fractionation methods that they were using or to the template they were using to capture the polymerase because you need something to capture it out of even of the fraction. Fractionation will also have hundreds of proteins now. You need to capture out the molecule. So maybe that the template they were using was not amenable or the fractionation method was not amenable and polymerase 1 was also very abundant. So all of these combinations together may have made this. Uh, but as I'm saying, this is the way the two other polymerases were identified was completely different. They could not do it with this fractionation method. They had to resort to mutations to find if there is no polymerase 1, then will the E. coli still replicate? Okay, now if it replicates, what is helping it to replicate? So again, they did another mutation and identified there is another mutation that stops it from replicating. So of those mutations, then they found the products were, some of them were polymerases, polymerase 1, polymerase 2, polymerase 3, because you could have helicases that can stop replication too. You could have other proteins that could stop replication too, but two of those turned out to be polymerases.
Yes, ma'am.
Absence of those bands. So they had to do some enormous amount of work to get to the other two polymerases.
Yes, ma'am. As it seems, yes. Thank you, ma'am.
Thank you. Thank you so much. We will carry on with another experiment on DNA polymerase 1, single molecule DNA polymerase 1, DNA polymerase activity, to see how what other techniques could be used to check for the motor activity of these polymerases. So once we have seen the processability, we have seen how its rate could be analyzed. We will see some other functions of it, how the exonuclease function and polymerase functions work. We are going to see that in the next class. All
Yes,
right.
ma'am.
Thank you. Thank you very much. For
Yes.
your exams, remember you will have a 3-hour exam. Ideally, you should be able to finish it in 1 hour. I'll give you additional 2 hours to finish it. Open book, open source, open internet, give it from any place, but please do not copy.
And ma'am, it would be after the minor, right?
Huh?
I mean, the syllabus would be after the minor. I mean, from the
Yes, yes, after minor. Syllabus will be everything that was taught after minor.
okay, ma'am.
So
Thank you.
up to major was up to minor was your kinases, kinesins, sorry, and anything after kinesins, dyneins, etc. Everything.
Yes, thank you, ma'am.
Okay, thank you, thank you, thank you very much. Thank you. Bye bye.
Bye.